morning, students. Today I'm going to continue with the host microbe interactions, and today we are going to discuss about the microbial virulence factors. Today we are going to discuss about the microbial virulence factors. And what is virulence? We have discussed in our last lectures of the host microbe interactions. So the virulence factors. So the virulence factors refer to the properties or basically the gene products that enable the microorganism to establish itself on or within a host of a particular species and enhance its potential to cause a disease. And these virulence factors are responsible for the virulence of the pathogen. Okay, so simply the virulence factors can be also defined as the factors are the properties of the microorganism which are produced by them. Okay, it may be within the host or maybe even outside the host. Okay, such as toxins. Botulism toxin may also produce outside the host. Okay, and generally some of them are even specific to a particular species, which enhance its potential or ability to cause a disease. Okay, and some of the general examples of the virulence factor include bacterial toxins, cell surface proteins that mediate bacterial attachment, and the cell surface carbohydrates and proteins that protects a bacterium, and hydrolytic enzymes that may contribute to the pathogenicity of the bacterium Okay, so how bacterial pathogens damage the host cells. They damage the host cells by using host nutrients. Okay, sometimes for example some bacteria may produce siderophores which enhance their capability to absorb iron. Okay, so due to which the cells of the body may get deprived from the iron. Okay, and second one is by causing a direct damage in the immediate vicinity of the infection. Okay, it may be done by various enzymes. Okay, next one is by producing toxins which are basically transported by blood and lymphatic system that damage the sites far removed from the invasion. Okay, and the last lectures we studied about how these are transported. Okay, once they enter lymph system and or blood, okay, they spread throughout the body, and sometimes by inducing hypersensitivity reactions Okay, so basically some of the invasion, okay, invasion hypersensitivity reactions, there are many systems also. Okay, simply let us discuss about the invasion. Okay, for example Shigella species basically initiate infection process by adhering to the host cells in the small intestine and there are multiple proteins including the invasion plasmid antigens, okay, that contribute to the process. And once inside the cells, the Shigella either are lynched or escape from the phagocytic vesicle where they multiply in the cytoplasm of the host cell. And other bacteria, okay, may invade a specific type of host epithelial cells also and may subsequently enter the tissue. And once inside the host cell, the bacteria may remain enclosed in a vacuole composed of a host membrane, composed of a host membrane, or the vacuole membrane may be dissolved and the bacteria may disperse within the cell from one cell to the another. So the invasion of the tissues followed by inflammation is basically enhanced by various factors, okay, which are we are going to write now is the virulence factors. Okay, virulence factors such as such as enzymes, antiphagocytic factors also. Okay, antiphagocytic factors also, biofilms, even how inflammation occurs, and the sorry as well as the intracellular survival, So this given here is just an overview of the virulence factors which a pathogen may have. Okay, not all the virulence factors seen here may be present in a single pathogen, but uh, it depends on the species and of the bacteria. Some may produce toxins, okay, some may have different types of enzymes, okay, some may have a capsule, okay, and some have fimbriae, some may have binding factors, some may have pili, and iron binding proteins which are siderophore or various other enzymes also. And for invasion they are going to use flagella or maybe toxins or enzymes such as kinases, coagulases, etc. Okay, so you can see each and every system has its own different type of virulence factors which are needed for the particular functions. Okay, for as example toxins, okay, their mechanism is different, adhesion mechanism is different, nutrient acquisition is basically absorption of nutrients from the host cell, invasion how they invade other parts of the body or other organ systems of the body, okay, and how they can avoid immune systems, okay, how they can avoid immune systems. Okay? Each and every part has a its own story. Okay, so starting with the microbial virulence factors, the first one are the enzymes. The enzymes basically invasion of bacteria is enhanced by various enzymes, and many species of the bacteria produce enzymes that are not intrinsically toxic but do play important role in the infectious process. Okay, some of the examples of the enzymes are hyaluronidases, collagenase, coagulase, streptokinase, etc. Okay, here are some of the examples of the enzymes. Okay, so starting with the first enzyme, which I have covered few enzymes. There are many other enzymes also. I will only cover few of them. For example, hyaluronidases and collagenase. Okay, so this hyaluronidases and collagenases are the enzymes that hydrolyze hyaluronic acid and degrade collagen respectively, okay, thereby allowing the bacteria to spread through the subcutaneous tissues. Okay, and some of the examples of the bacteria which may produce hyaluronidases are Staphylococci, Streptococci, and some of the anaerobes, and which aid in the spread of their tissues. For example, hyaluronidase produced by Streptococcus pyogenes degrades the hyaluronic acid in the subcutaneous tissues and thereby facilitating the organism to spread quickly, okay, or rapidly. Another example is of a Clostridium perfringens, which produce a proteolytic enzyme collagenase and which basically degrades collagen. Okay, what is collagen? It's a major protein of the fibrous connective tissue. Okay, it's a major protein of the fibrous connective tissue and promotes the spread of infection in the tissue. Now let's have a look at the diagram. Okay, Let's have a look at the diagram given here. Okay, in this diagram, okay, now this one here is a hyaluron, okay, hyaluronic acid, or you can say, okay, this here given is an hyaluron, okay, here, okay. So inside and the outside of the cell, okay. So basically hyaluronic acid or hyaluron ronan is basically a polymer which is found in layers of the epidermis that connects the adjacent cells, okay. And the enzyme hyaluronidase basically breaks this one, okay, breaks this layer, okay, breaks this layer, and it basically connects two cells, and there is another layer which basically is present here, which is made up of a collagen, which is made up of a collagen, okay. Now similarly to hyaluronidase, collagenase basically breaks this layer, okay. The bacteria produce enzyme collagenase and breaks the layer so that bacteria can invade deeper tissues, so that the bacteria can invade the deeper tissues, okay. So now moving forward with the next enzyme, coagulase. Next enzyme is coagulase. The staphylococcus aureus produces the enzyme coagulase, which in association with blood factors coagulates the plasma, okay. And the coagulation contributes to the formation of fibrin wall around the staphylococcal lesions, which protects the bacteria from the phagocytosis by walling of the infected area. And the enzyme also causes deposition of the fibrin on the surfaces of the individual staphylococci, which protects them from the phagocytosis or from the destruction within the phagocytic cells. Here fibrin acts as a barrier for this staphylococci, okay. Now please check here in the diagram, okay, where the enzyme coagulase, okay, which is produced by the bacteria, okay. And what will they do? They will form a clot around them, okay. And this clot basically helps as a layer for the staphylococci or other bacteria which produce this coagulase enzyme against various immune system mechanisms, okay. And later, okay, if they want to infect further, okay, they produce the enzyme kinase, okay. And this kinase basically dissolves the clot, okay. This kinase basically dissolves the clot and releases the bacteria in the cell layers Now next enzyme is the streptokinase, also known as fibrinolysin, okay. It's an enzyme which is also used in which was also used in medical treatment also, especially in the case of some diseases, okay, but it is not used nowadays, and but it is produced by a bacteria streptococcus. Which activates a proteolytic enzyme of the plasma, okay, which activates a proteolytic enzyme of a plasma. This enzyme is able to dissolve coagulated plasma and thereby possibly aids in the rapid spread of streptococci through the tissues. So whatever the plasma, okay, basically is present, okay, is broken down by this enzyme, is being broken down by this enzyme. And the streptokinase, as I told you, has been used in the treatment of acute myocardial infarction, okay, to dissolve the fibrin clots, to dissolve the fibrin clots, okay. Now please have a look here. And one of the enzyme responsible for the rapid spread is the streptokinase. And this strains that produce streptokinase digests the fibrin clot that the body uses to isolate an infection. And normally, body produces plasmin to break down unneeded blood clots. And the streptokinase breaks down the body's precursor plasminogen, breaks down the body's precursor plasminogen to produce plasmin. Okay. Now streptokinase, which is produced by the bacteria, okay, which is produced by the bacteria, when combines with the plasminogen, it converts plasminogen into plasmin. Okay, and this plasmin basically breaks down the fibrin. Okay, thereby dissolving the layer which acts as a protective layer for the body in order to stop the infection is being broken down by the enzyme streptokinase. Okay, now okay, you can see here a small child who is infected with a bacteria, and this bacteria is producing very high amount of the streptokinase, is producing very high amount of the streptokinase. Okay, this is given here. It's a very serious effect of the enzyme streptokinase. Now moving forward with the IgA1 proteases. IgA1 proteases. Certain pathogenic bacteria produces enzymes IgA1 proteases that split IgA1 at a specific proline threonine or proline serine bonds in the Hind region and inactivate its antibody activity. Okay, inactivates its antibody activity. This is how IgA1 basically works, and it is produced by many of the bacteria and is one of the important virulence factor of the pathogens such as Neisseria gonorrhoeae, Neisseria meningitidis, Haemophilus influenzae, and Streptococcus pneumoniae So the production of the IgA1 protease allows the pathogen to inactivate the primary antibody found in the mucosal surfaces and thereby facilitates its attachment to this bacteria to the mucous membranes. Okay, so they are going to inactivate the primary antibody. For example, let us assume this is an antibody. Okay, this one is an antibody and this is an IgA1 proteases. Okay, this is an IgA1 proteases. So what will they do? They will simply bind with the IgA antibody, and thereby limiting its functions. By inactivating the antibodies. Okay. Now moving forward with the antiphagocytic factors. Okay, where many bacterial pathogens are rapidly killed once they are ingested by polymorphonuclear cells or macrophages. Okay, through phagocytosis, most of the pathogens are killed, but some can survive. Okay, and evade this phagocytosis or leukocyte microbicidal mechanisms, and by several antiphagocytotic factors, okay, such as capsules, soluble proteins, cytotoxins, as well as the surface antigen can prevent this phagocytosis. Okay. So this basically this production basically of these factors basically aid the pathogen to survive a longer time in the host. So starting with the capsules, okay, starting with the capsule surrounding the bacteria, such as in the case of Streptococcus pneumoniae and Neisseria meningitidis, okay, is the important antiphagocytic factor, and it retards phagocytosis of bacteria by preventing the phagocytes from adhering to the bacteria, by preventing the phagocytes, okay, from adhering or attaching to the bacteria. Okay, now next one is a cell wall protein. Next one is a cell wall protein. The cell wall proteins such as protein A and protein M of Staphylococcus aureus and Streptococcus pyogenes especially are the antiphagocytics. For example, protein A of S. aureus binds to the IgG and prevents the activation of the complement. Okay, so protein A of Staphylococcus aureus binds to the IgG and prevents activation of the complement. You all know what is complement system. You have already studied, okay, in your previous units of this immunology. The M protein of the Streptococcus pyogenes is antiphagocytic. Okay, next comes the cytotoxins. Certain bacteria produce cytotoxins that interfere with the chemotaxis or the killing of the phagocytes. For example, Staphylococcus aureus produces hemolycins that lyse and damage RBCs. Okay, that lyse and damage RBCs and WBCs. For example, hemolycins basically lyse the RBCs, and leukocidins basically damages the WBCs, thereby interfering with their function. Okay, the next comes the surface antigens. Surface antigens of the bacteria such as VI antigen of the Salmonella typhi and the K antigen of the E. coli makes the bacteria resistant makes the bacteria resistant of the E. coli makes the bacteria resistant makes the bacteria resistant to phagocytosis and the lytic activity of the complement. Okay, so in the case of Salmonella, VI antigen, and the case of E. coli, okay, E. coli basically resists the phagocytosis and the lytic activity of the complement. Okay, now how this phagocytic factors help. So, escaping the phagocytosis, pathogens have evolved many ways to escape phagocytosis. Okay, one of them is by the hydrolytic enzymes. Now, given here is an phagocytosis. It's an sorry. This one is a regular diagram of a phagocytosis. Okay, now what are these pathogens going to do? Okay, what are these pathogens going to do? One of the technique is by killing the toxins. Is by releasing the toxins which kills the phagocytes. Okay, for example, this bacteria produces toxins and kills the phagocytes. Okay, second one is by producing a capsule. The second one is by producing a capsule. Okay, and through which it can avoid phagocytes. Okay, through which it can avoid phagocytes. Now moving further, the next one, by blocking the fusion of the lysosome with the phagosome. By blocking the fusion of the lysosome, okay, with the phagosome, okay. So they are going to basically block the fusion of lysosome with the phagosome, okay. Next one is by escaping next one is by escaping phagosome and living in a phagocytic cell okay, as many of the pathogens do have capability to live inside the phagocytic cell, okay. Now moving forward, next one is by adapting to a harsh environment of the phagolysosome and neutralizing the hydrolytic enzymes, okay. Even if it is further grows into a phagocytic cell, it has a capability to adapt to this environment. Some of the bacteria may have capability to adapt to this environment, okay. Next one of the microbial virulence factor is the biofilms is the biofilms. A biofilm is an aggregate of interactive bacteria attached to a solid surface or or to each other encased in an exopolysaccharide matrix. In this case, exopolysaccharide will act as a protective layer to this bacteria against the host immune defense systems, okay. So biofilms consisting of a single cells and microcolonies of the bacteria all found together in a highly hydrated and predominantly anionic exopolymer matrix, okay. And this is a distinct from the planktonic or free living bacterial growth, in which the interactions of the microbes do not occur. So the biofilms form a slimy coat on the solid surfaces and occur throughout the nature, okay. And a single species of bacteria may be involved, or more than one species of bacteria may also co-aggregate to form a biofilm. And biofilms are important in human infections that are persistent and are difficult to treat, okay. And few examples of the infections include Staphylococcus epidermidis and SOS infections of the central venous catheters, okay, okay, catheters, uh, okay. And the next one is a eye infection that occur in the contact lenses and the intraocular lenses. Next one is the infection in the dental plaque, okay, the plaque which occur in our dental cavities. And next one is the Pseudomonas aeruginosa airway infections in cystic fibrosis patients, okay. So this is how biofilm basically are involved in infections. So for your information, what is catheter, okay. Catheter is just a soft tube like structure. It is an artificial one, okay. It's basically passed into the bladder to basically drain the urine out, okay. And it is used especially if you have operated recently, okay, in order to empty the bladder, okay, okay. Now moving forward with the photographs. Okay, now you can see here in the catheter, the the surface of the catheter. Okay, and the green one here is an biofilm which appears green in color. Okay, and after some time, okay, if it remains untreated, most of the bacteria producing slime are not visible in the biofilm, where you can see now slime has been covered. Okay, slime has covered most of the bacteria and also the catheter, and this cannot be easily be treated. Okay, so this slime producing bacteria are most common causative agents of infection in the indwelling devices, okay, which are used after a operation in some of the cases, okay, in order to drain out some of the liquids, and they adhere to the surfaces such as plastic catheter, which is given in the photo. Okay, in which A basically they begin to divide as I told you, and in the B where the biofilm has almost covered most part of the bacteria. Okay, now comes the other microbial virulence factors, okay, in which inflation. So you already studied infection. I'm not going to go in more detail about the inflammation right now. So inflammation is an important host defense induced by the presence of the bacteria in the body, and it is of two types when it comes to infection. In the case of an infection, pyogenic and granulomatous. Okay, the pyogenic infection is the host defensive primarily against the pyogenic or pus producing bacteria such as Streptococcus pyogenes, and it typically consists of a neutrophils and the production of specific antibodies and the elevated level of the complement. So this pyogenic inflammation is basically present in the case where pus is being produced by the bacteria or by the bacterial infections. Okay, next comes the granulomatous inflammation. Granulomatous inflammation is the host defense seen primarily against intracellular granuloma producing bacteria such as Mycobacterium tuberculosis. Okay, it has capability to produce granules, okay, inside the respiratory system. Another one is a Mycobacterium repare. Similar, it has also capability to produce granulomatous inflammation, and the response consists of the production of macrophages and CD4 T cells, CD4 plus T cells. Okay, now next one is the microbial virulence factors, in which includes the intracellular survivance. Okay, a few mechanisms that are suggested for the intracellular survival is the first one is the inhibition of phagolysosome fusion. Second one is the resistance to lysosomal enzymes, and third one is the adaptation to cytoplasmic replication. Third one is the adaptation to the cytoplasmic growth. Okay, so the bacteria such as chlamydia, mycobacterium tuberculosis basically interferes with the formation of phagolysosomes in a phagocytes that can survive intracellularly, and these bacteria live within the cells and are protected by the macrophages and neutrophils, and these bacteria do not interfere with the formation of phagosomes and are otherwise killed inside the phagocytes. Okay, so this bacteria, okay, can interfere with the formation of a phagolysosome phagosomes. Okay. And another example is the presence of the polysaccharide in the Mycobacterium leprechaunium and the mycosite in the Mycobacterium tuberculosis makes this bacteria resistant to the action of the lysosomal enzymes. Okay, and the next one is a certain bacteria such as Rickettsia escape the phagolysosome into the cytoplasm of the host cell before the fusion of the phagosome with the lysosome takes place. Hence continue to remain intracellular. Okay, as these phagocytes can sometimes not detect the intracellular pathogens. Okay, so this one are some of the examples of the frustrated phagocytosis where the phagocytosis is not able to move forward. So in this case, first one, a planktonic cells are sensitive to the antibiotics and are detected by the host phagocytes and the antibodies. Now here you can see where a detection occurs. Okay, where a detection occurs of the antibacterial antibodies are also present. Now in the second one, the planktonic cells basically settle down. Now you can see the settling of the planktonic cells settle down to form a biofilm and become biofilm cells given here. Okay, this one is a biofilm which is formed. Okay, and at the same time, planktonic bacterium has been also taken up by the phagocyte. Okay, now in the third scenario, what happens? The biofilm cells resist the antibiotics. Okay, this biofilm basically is going to resist the antibiotics and even the antibody detection. Okay, and the host phagocytes detect it and attempt to destroy biofilm cells biofilm cells. Now in the D1, this in this case unable to capture biofilm cells. Okay, unable to capture the biofilm cells, the phagocyte releases the antimicrobial products. Okay, that basically kills the host cells. But not the biofilm cells. This is how uh, pathogens basically evades the phagocytosis. Okay, so please join the webex session. Okay, so everyone please join the webex session. I'm going to end the lecture. Please join the webex session.